So thanks everybody for being here. The title of my talk is The Impact of Transitive Annotation on the Training of Taxonomic Classifiers. And just to give you a very quick high-level idea of what we're trying to talk about is the fact that when you are doing some sequencing experiment, you get a sequence, you try to figure out what the sequence is by using a classifier, then put the sequence in a database with that label. And now the question is what happens if another classifier trains on that sequence that itself was generated by a classifier, right? But before I go into my talk,
Your audio's off.
oh, my audio's off. I apologize.
That's not my fault, probably. All right, I'm not. Or not. My bad.
Okay, we good?
Yeah.
It's on there.
Okay. I think.
Okay, yeah, so we're good. Before I go further, I want to authors, so Hari, who did the bulk of the work, he recently graduated with his PhD. He's at Latch Genomics in California. Noam Fox was an undergrad student, and she just graduated and followed a career as a postbac, I think, trying to do an MD PhD. And Athalie Bonin is a PhD student who was actually at the meeting, so you can actually hopefully run into her, and she has a poster. Right, so I want to come, so I set up the problem. I want to come to the question of how reliable databases really are. So this is from a paper from Jana Bromberg. Jana is also at the conference, and you can chat with her about her paper. But she pointed out that in SwissProt, which is one of the highest quality protein databases that we have, there are about 214,000 proteins with enzyme commission numbers. So these are the enzymes of the database, and only about 7,000 of them have experimental evidence for them. It's interesting to read her paper because one of the sections is called functional annotations, even in manually curated databases, are frequently incorrect. And the second observation is that there are EC numbers that exist in a database, but there's no experimental evidence for any sequence in that EC number, right? So essentially, it's kind of annotations that just pop up from nowhere. And this is a surprise. Well, I found a paper from 1998 that essentially makes the case that because we have so much data, so this is in 1998, we had too much data. This is not the new thing when all our talks that we have too much data today. But essentially what the idea was we have too much data, computers need to analyze that data, but computers can also make errors. So we knew about this in 1998, things have not improved since then. So we set out to try to understand more precisely what's going on. Okay, we note that as a problem, but we don't have a way of actually quantifying what the problem is, what the extent of the problem is, and so essentially we can't really do anything if we don't have some theory or at least documented information about what happens. So we have set up this setting. So we have a classifier. In our case, we use the RDP classifier, so it's a naive-based classifier used primarily in 16S ribosomal RNA sequencing. It's very heavily used. It's one of the most commonly used tools, right? That classifier depends on a database. That's the training data for the classifier. So we take two sequences, sequence 1, sequence 2. We run them through a classifier. We get labels, which I call label 1 for each of these, which is the label produced by a classifier 1, right? Now, our question is, what if we take that sequence 2 and add it back to the database, right? So now, essentially, I have a new database that just has sequence 2 added to it. It's just one single sequence in our experiment, right? And that sequence with its label becomes a new database. We retrain the classifier. Now we have a second classifier. Once we've retrained the classifier, we can reclassify sequence 1. And the question is, how different is the label 2 of sequence 1 from label 1 of sequence 1, right? How much did the classification change because I changed a single sequence, right? Ideally, not much happens, but that's what we set out to find out. I will skip this description of how the RDP classifier goes, but primarily it just counts k-mers and does a bit of statistics. The one observation that we had, and we tried to figure out essentially what happens, like how do we find out where the labels change? If I have a sequence with one label and another sequence with another label, what does the space the classifier is creating, this virtual space that it's creating to decide for a new sequence, does it classify with sequence 1 or sequence 2? So the way we've done that, we essentially take two sequences with the divergent labels. we compute an edit path between them. So essentially, we create artificial sequences that each differ by one base from each other until we convert sequence 1 to sequence 2. And then we classify those sequences and see where the label changes. What does this look like in reality? These are 10 different ways of actually computing that edit path. The order in which you make the edits matters, right? There could be a lot more, but on the left, you have a sequence called Alicinella. So we call it all sequences labeled Alicinella yellow. On the right, there's a sequence called Dialister, colored all these intermediate sequences in blue if they match the Dialister. And then the gray underneath is the confidence the classifier has that particular label gets assigned to that sequence, right? So that's what those paths look like. That's what that decision boundary looks like, at least for these two sequences. And you can see the boundary is not exactly in the same place depending on which place the classification happens. Like this space is highly dimensional, complex space. We're simplifying it a little here, right? So we did a very simple experiment. We said, well, what's the worst case that could happen? We take a sequence that's right on the decision boundary, and we add it to the classifier with the label that it has, right? So essentially, that's that yellow sequence. We add it back to a classifier, says we believe this is yellow, and then retrain the classifier with that sequence. Once we do that, what happens? All the sequences get classified as Alicinella, all of them. Not only that, if you see the gray, it's high. So essentially, the classifier is not only changing all the labels, but it's very, very confident in those labels, right? So this is clearly a problem. Now the question is, well, what's the extent of the problem? And that's where we kind of delved into a couple other questions. So one of our questions was, how far do you need to be? Like this, I would say adversarial example, how far does it need to be for a decision boundary to cause a problem? And you can see that the further away you go, the less of a problem it is. The height is how many sequences change labels because of it. We asked the question of which sequences are more fragile. Like if I only have a representative from Alicinella in the database, perhaps it's easier to change that than for example, a sequence like E. coli when there's thousands of sequences that are labeled E. coli. Perhaps that's harder to modify, right? And we've looked at both ends, like both the sequence that we're trying to change the labels to and the sequence for which the label gets changed. We've also looked at whether we need to create any artificial sequence or just use real sequences. And it turns out on the left are the real sequences. The impact is lower, but there's still an impact, right? And also try to figure out, well, what if can I delete a genus from the database by simply adding more and more adversarial examples? We were not able to delete a genus. So essentially the number of sequences you see, the impact plateaus, but there's a significant drop. I can remove a lot of labels from the database corresponding to real sequences. And there's some all sorts of interesting observation there that I won't have time to go into, but I can discuss further in our paper. Some takeaways here is I think the naive-based classifiers might be uniquely vulnerable. So perhaps other classifiers are more stable, but no one has looked into this, and I think it's important that we start looking at like how do hidden Markov models react in this mode. And I feel that there's a lot more work that needs to happen, both in particularly in functional annotation. So I think that's also a big thing. Taxonomy, I think, doesn't matter that much because it's all made up anyway. But functional information is very important, right? And I think it's important to look into that and see, well, are the databases of functional annotation also vulnerable to this kind of phenomena? So that's it. Thank you. And I'm not sure if I have time for a question because we have a next speaker, but thank you so much for your attention.